Hello everyone. In continuation of chapter 8 Human Health and Disease, we will now talk about one of the most dreaded diseases of human beings, that is cancer. Has affected number of people all over the globe. More than 1 million Indians suffer from cancer and large number of them die of cancer every year. Now let's understand what is a cancer. Cancer is an abnormal and uncontrolled division of cells known as cancer cells. These cells can invade and destroy the surrounding tissues. Generally, a cancer can be defined as an uncontrolled proliferation of cells without any differentiation. In our body, cell growth and differentiation is highly controlled and regulated. Normal cells show a property or a regulatory mechanism known as contact inhibition. Contact inhibition is a mechanism that functions to keep cells growing into a layer one cell thick only. That is, it will make the cells grow into a monolayer. If a cell has plenty of available space around, so for example, this cell goes missing, the other cell will replicate quickly and will move freely into this space. With this property, normal cells contact with other cells and inhibit their uncontrolled growth. In contrast to the normal cells, cancer cells have lost the property of contact inhibition. These cells proliferate in an unregulated manner and they form clones of cells which can expand regularly. So this uncontrolled growth of cells is called as tumor or neoplasm. Now let's differentiate between a normal and a cancerous cell. Normal cell shows contact inhibition, whereas such phenomenon is not shown by cancer cell. Cancer cells are non-specialized cells. They do not show any controlled growth mechanisms, whereas normal cells are specialized cells which show controlled growth. Normal cells have normal chromosomes, whereas abnormal chromosomes are seen in cancer cells. Every normal cell is having a definite lifespan and it also shows apoptosis, that is natural cell death. Cancer cells do not have definite lifespan and no apoptosis is shown by cancer cell. Cancer cells lead to the formation of tumors. Tumors are of two types, benign tumors and malignant tumors. Benign tumors are the ones which will remain confined to their original location only, and these will not spread from one part of the body to another. Thereby, these tumors they cause very little damage. Their rate of growth is usually slow and they do not show metastasis. We will learn about metastasis in the next slide. These types of tumors are generally not considered cancerous. The malignant tumors, on the other hand, are a mass of proliferating cells which are called as neoplastic or tumor cells. These neoplastic or tumor cells can invade normal tissues, thereby causing damage to them. Cells of malignant tumor grow very actively and rapidly and show metastasis. Malignant tumor are considered cancerous. Now let's understand what is metastasis. Cells of malignant tumor, that is primary tumor, actively divide and grow. They will starve the normal cell by competing them for the vital nutrients. Cells which are slowed off from such tumors will enter the bloodstream. From the bloodstream, they will move to the distant sites. Wherever they will get lodged, they will start dividing there and will lead to the formation of a new tumor, that is a secondary tumor will be formed. So this property of the cancerous cells to move from one place to other through some body fluids and resulting into the formation of secondary tumors is said to be metastasis. Metastasis is the most feared property of malignant tumors. Causes of cancers. Number of substances or agents are responsible for the cause of cancer. The physical or chemical agents which lead to the cancers are called as carcinogens. There could be physical irritants, for example, heat can cause cancer. Kashmiris use kangri near their abdomen, so this could lead to the skin abdominal cancer. And the excessive exposure to UV radiations, that is the sunlight, can also cause skin cancer. Tobacco chewing can lead to oral cancer. Smoking tobacco could lead to lung cancer. There are a number of chemical agents also which are causing cancer. These may include caffeine, nicotines, products of combustion of coal and oil, pesticides, and constant use of artificial sweetener can cause cancer. An animal protein rich diet can also cause cancer of large intestine. Some viruses and other parasites are also responsible for cancers, and sometimes excessive secretion of hormones could also lead to cancer. Radiations, ionizing radiations like X-rays and gamma rays, and non-ionizing radiations like UV rays can cause permanent damage to DNA, thereby leading to cancer. There are certain viruses which are containing viral genes. These are oncogenic viruses which could lead to cancer. It is noted that normal cells are having certain genes called cellular oncogenes or proto-oncogenes, which when activated under certain conditions could change into oncogenic cells. Jumping genes or transposons are often involved in this type of conversion. Some of the important sites of cancer are skin, mouth, esophagus, stomach, colon, rectum, liver, gallbladder, pancreas, blood, lymph, adipose tissue, lung, uterine cervical cancer, breast, brain, penis, prostate cancer, muscles, thyroid, kidney, and bones cancer. Possible symptoms of cancers include persistent cough or hoarseness in a smoker. There could be persistent change in digestive and bowel habits. There could be change in a wart or a mole, unexpected diminished or loss of appetite, unexplained low-grade fever, loss of weight which is unexplained, any ulcer which is uncurable, bleeding in vagina at times other than menstruation, non-injury bleeding from the surface of the skin, mouth, or any other opening of the body. These types of symptoms can also be present in a normal individual, but if these symptoms have turned persistent and have become uncurable, one should immediately see a doctor. Cancer detection and diagnosis is possible if a cancer is detected at early stages in an individual, so that individual can be survived from cancer. Cancer detection is based on biopsy and histopathological studies of the tissue and blood. In biopsy, a small piece of suspected tissue is taken and it is cut into thin sections. It is stained and then it is examined under the microscope by a pathologist. If a patient is suffering from leukemia, that is blood cancers, bone marrow tests can be done for the diagnosis. These tests can help to count the increased numbers of cells in the patient. Some techniques like radiography, which uses X-rays, computed tomography, CT scans, magnetic resonance imaging, MRI, are also used to detect the cancers of internal organs. Computed tomography uses X-rays to generate three-dimensional images of the internal organs of an individual. Magnetic resonance imaging uses strong magnetic fields and non-ionizing radiations to accurately detect pathological and physiological changes in the living tissue. Magnetic resonance imaging uses strong magnetic fields and ionizing radiations to accurately detect pathological and physiological changes in the living tissue. Antibodies against cancer-specific antigens are also used for detection of certain cancers. like monoclonal antibodies coupled to appropriate radioisotopes can detect cancer-specific antigens and hence cancer. With the advancement of techniques in molecular biology, genes can be detected in individuals with inherited susceptibility to certain cancers. Identification of such genes which predispose an individual to certain cancers may be very helpful in prevention of cancers. Such individuals may be advised pre-hand to avoid exposure to particular carcinogens to which they are susceptible. For example, if a person is susceptible to the incidence of lung cancer, he can be advised to avoid tobacco smoke. Cancer can be treated. 4 different approaches are used. First is surgery, radiation therapy or radiotherapy, chemotherapy, and immunotherapy. Surgery involves the removal of the entire cancerous tissue. In radiotherapy, tumor cells are irradiated lethally and it is taken care that normal cells which are surrounding the tumor mass are not destroyed. Several chemotherapeutic drugs are also used for killing the cancerous cells. Some of these are specific for particular types of tumors, but many of these drugs have side effects. The patient may suffer hair loss or anemia. Under immunotherapy, it involves natural anti-cancer immunological defense mechanisms. Monoclonal antibodies are used in various ways. Example, radioimmunotherapy is used for treatment of cancer. Sometimes the cancer cells escape the immune system of the patient. So therefore, the patients are additionally supplied with biological response modifiers such as alpha interferons. These interferons will activate their immunity system and help in destroying the tumors. Children, I hope you have understood the basic concept of cancer, its causes and various approaches of treatments. Now let's see whether you can answer these questions. Differentiate between a normal cell and a cancer cell. Define metastasis. Name 2 techniques which are useful to detect cancer of internal organs. Why are cancer patients often given interferon as part of the treatment? What are tumors? Describe its types. Thank you and have happy learning.